transgenic animals and the legal and social economic impact of them. Introduction. A transgenic animal is one that carries a foreign gene that has been deliberately inserted into its genome. The DNA introduced is called a transgene and the overall process is called transgenesis. Foreign genes are inserted into the germline of the animal so it can be transmitted to the progeny. Transgenic technology has led to the development of fishes, livestock and other animals with altered genetic profiles which are useful for mankind. The foreign gene is constructed using recombinant DNA technology. In addition to the structural gene, the DNA usually includes other sequences to enable. First transgenic animal. First transgenic animal was a super mouse created by Ralph Brinster and Richard in 1982. It was created by inserting a human growth hormone gene in the mouse genome. The offspring was much larger than the parents. You can see the comparison in the picture. Mouse is the common transgenic animal for experiments. Other animals include pig, goat, cow, sheep, fish, etc. What is the methodology to create transgenic animal? Step 1 is construction of a transgene. Transgene will be made up of 3 parts, that is a promoter and gene to be expressed and the termination sequence. Step 2 is introduction of foreign gene into the animal. It is either by pronuclear microinjection method or embryonic stem cell method. These transfection methods are discussed in your earlier session. Step 3 is screening for transgenic positives. That is, transgenic progenies are screened by PCR method to examine the site of incorporation of the gene. Some transgenes may not be expressed if integrated into a transcriptionally inactive site. The gene expression is found out by PCR technique by the use of marker gene. Then the step 4 is further animal breeding is done to obtain maximum expression. So pronuclear microinjection method. We'll have the overview. A female animal is superovulated and eggs are collected. You can see in the picture. The eggs are then fertilized in vitro. The transgene containing solution is then injected into the male pronucleus using a micropipette. Then eggs with the transgenes are kept overnight in an incubator to develop to a 2-cell stage. Then the eggs are implanted into the uterus of a pseudo pregnant female, that is the female which has been mated with a vasectomized male the previous night. Then in this way the transgenic mouse is formed by pronuclear microinjection method. Next we have embryonic stem cell method. Transgenic animals can be created by manipulating embryonic stem cells. Here embryonic stem cells are obtained from the inner cell mass of a blastocyst. Transgene is incorporated into the embryonic stem cells by microinjection or by a retrovirus method or by electroporation. All these transgenic methods, transfection methods, we have studied in the earlier session. So either of these methods, the transgene is incorporated into the embryonic stem cell. Then transgenic stem cells are grown in vitro. You can see in the picture the stem cells are grown in vitro. Then they are inserted into a blastocyst and implanted into a host uterus. Okay, cells expressing desired genes are selected. Then the transformed genes, embryonic cells, are injected into the inner cell mass of the blastocyst. Then this blastocyst is implanted into the uterus of the host animal, or we call them as surrogate mother or foster mother. Then the offsprings which are produced are tested for presence of the particular gene we have inserted. Then heterozygous mating of offsprings are done in order to produce homozygous transgenic strain. Transgenic mice. I have already told you about supermouse, that is it contains a human growth hormone. Next one is transgenic mice expressing the human gene for the poliovirus. Poliovirus receptor gene is expressed in transgenic mice so that it can be infected by poliovirus and develop paralysis. So it serves as a disease model. So to study the poliovirus disease, people are using transgenic mice, but they lack in the receptor, that is poliovirus receptor gene, so that it cannot identify the poliovirus. So human gene for the poliovirus receptor has been introduced into the mice, and it is made as transgenic mice, so that the transgenic mice which has poliovirus receptor will develop the symptoms of polio disease, that is paralysis, etc. So it serves as a disease model for that particular disease study. Next is this also serves as an inexpensive model to study that disease. Next is Alzheimer's mouse. In the brain of Alzheimer's patients, dead nerve cells are entangled in a protein called amyloid. Okay. Amyloid is a protein that is entangled in the dead nerve cells of brain of Alzheimer's patients. Okay. So the mouse made by introducing amyloid precursor gene into the fertilized egg of mice in order to make Alzheimer's mouse. Again, this is also a disease model. Oncomouse. The oncomouse is a strain of transgenic mouse containing a human gene that causes breast cancer. Okay, it is made by inserting activated oncogenes. This enables the researchers to study clearly the development of cancer. Transgenic cattle. The transgenic cattle, that is cow containing a bovine alpha S1 casein promoter. So here the oocytes obtained from the ovaries of the slaughterhouse cows were cultured and fertilized in vitro. Their pronuclei were microinjected with a DNA construct containing a gene, that is bovine alpha S1 casein promoter, driving the expression of the human iron binding protein, that is lactoferrin. Okay, the embryos were cultured till the blastula or morula stage and then non-surgically transferred to surrogate mothers. Only 2 of the 19 calves were born from 1 or 3 zygotes were transgenic. Okay, only 19 calves were transgenic from 1 or 3 zygotes. One male and other was female. Then hepatitis B surface antigen. In 1998, transgenic cows carrying a gene for hepatitis B surface antigen were produced through the transfer of gene to unfertilized oocytes arrested at metaphase. Then antibody production. A human artificial chromosome containing the complete sequence of the human immunoglobulin, both heavy and light chain loci, was introduced into bovine fibroblasts, which were then used in nuclear transfer. The transchromosomal bovine offspring were obtained that expressed human immunoglobulin in their blood. This system could be a significant step forward in the production of human therapeutic polyclonal antibodies. Dairy industry. Dairy production is an attractive field for targeted genetic modification. The bovine casein ratio has been altered by overexpression of beta and kappa casein. This clearly showing the potential for improvements in the functional properties of bovine milk. This has shown up the improvements in the properties of bovine milk, that is beta and kappa casein gene. It may be possible to create hypoallergenic milk by knockout of the lactoglobulin gene. To generate lactose-free milk by a knockout of the lactoalbumin, that is A-lactoalbumin locus, which is suitable for consumption by many lactose intolerant persons. Transgenic fish. Superfish. To get increased growth and size in the fish, growth hormone gene was been inserted into fertilized egg. Then the transgenic fish grows about 10 to 11 times faster than normal fish. That is, in case of salmon fish, transgenic salmon grows about 10 to 11 times faster than the normal fish. These growth hormones have been inserted into the golden fish also. Um, Around 35 species of fish have been about 35 species of transgenic fish have been developed. Some of the transgenic fish contain genes taken from other fish, humans, and insects also. Examples are salmon, uh, tilapia, trout, catfish, flounder, goldfish, and oysters. Glowfish, genetically modified. It is a genetically modified freshwater zebra fish. The scientific name is Denio rario, commonly known as zebra fish. It is a freshwater fish that is produced by inserting a fluorescent protein gene. We say C anemone gene for fluorescent protein gene that is taken out from jellyfish, and then it is inserted into the embryo of zebrafish in order to get that glowing fluorescence colors in the pet fishes. Environmental pollution indicators. Some examples include fish carrying metal responsive or heat shock promoters spliced into green fluorescent genes again. Okay, in order to provide an in vivo indicator of exposure to heavy metals or other pollutants. If they come across or exposed to heavy metals or other pollutants, the gene inside it will make them fluoresce. So they can be used as pollution indicators. Transgenic pig. Enviro pig. Pigs have trouble in fully digesting a compound known as phytate, which is found in many cereal grains used to feed them. Okay, the transgenic pigs are created by introducing phytase gene, which is taken out from E. coli. Okay, pigs have trouble in digesting a compound that is phytate, so the phytase gene from E. coli is taken, and that phytase enzyme, that gene is introduced into the pigs to get the transgenic pigs. So the offspring which is developed, that is a transgene animal, in that animal we can find phytase enzyme which is produced in the salivary gland of the transgenic pig. So it helps to degrade indigestible phytate with the release of phosphate that is readily digested by pigs. Okay. Then pig for organ transplantation. Yes, we'll also call it as xenotransplantation. So, organ transplantation technology has led to an acute shortage of appropriate organs. um, of our live organ donation does not meet the demand in Western societies. So, to close the growing gap between demand and availability of appropriated human organs, xenografts from transgenic pigs are considered to be the best alternative. Although there was a problem of rejection, so now that has been um, cleared by introducing human genes into the pigs in order to decrease the chance of organ rejection by the human body. Next is transgenic sheep. Uh, in July 2000, there was a success in inserting a transgene into a specific gene locus was reported. The gene was the human gene for alpha 1 antitrypsin. Alpha 1 antitrypsin. And two of the animals expressed large quantities of the human protein in their milk. It is used to treat the patients of emphysema. Anti-hemophilic factor. Roslin Institute, which created Dolly in 1996, a year later they produced two more clone shapes called Molly and Polly, which were having a human anti-hemophilic factor gene in their genome. They secrete the human clotting factor 9 in the milk, which can be purified and administered to the hemophilic patients. Higher wool production. Genes essential for the synthesis of some important amino acids found in keratin proteins of wool have been cloned and introduced into the embryos to produce transgenic sheep. Genes cis-E coding for serine acetyltransferase and gene cis-M coding for O-acetylserine sulfhydrolase isolated from the bacteria were cloned into a vector and later introduced into sheep cells. Sheep with growth hormone gene have been introduced into sheep, which promotes the body weight of the sheep, thus they yield more mutton. Transgenic goat. Transgenic goats engineered to produce tissue plasminogen activator. This protein is used for treatment of coronary thrombosis. Then goats producing spider silk in milk are also produced. Spider silk is stronger than steel, elastic and extremely lightweight. However, unlike silkworms, spiders cannot be farmed for their silk. There is a company called Nexia Biotechnologies that has been successful at producing spider silk in the milk of goats and is in the process of developing BioSteel, a man-made fabric made of spider silk. The potential applications of this fabric are from medical uses, wound closure, dressings, patches, glues, etc. For military uses, it is a strong lightweight body armor. In sporting goods, biodegradable fishing line. So these are all the applications estimated in spider silk. Then transgenic goats engineered to produce human breast milk. University of California scientists created a transgenic goat by transferring human genes for breast milk enzymes and proteins into goat embryos. They produce 60% of the lysozyme and lactoferrin found in human mother's milk. So, for the babies of mothers who aren't present or can't nurse them, for them, milk from these transgenic goats could provide the best alternative. Transgenic monkey. Andy was the first transgenic monkey which was born in 2000. Andy stands for inserted DNA. Inserted DNA, that is spelled backward. AND, inserted stands for I. So ANDI. Andy is the name. Oregon researchers have created the first genetically modified monkey, that is Andy. Rhesus monkey, it was a playful rhesus monkey born on October 2nd, 2000, has been engineered to carry a gene, that is green fluorescent protein, taken from the jellyfish, which glows when exposed to UV light. Okay. Andy proves that transgenic primates can be created and can express a foreign gene delivered into their genome. They inserted the unfertilized monkey eggs with a green fluorescent protein transgene using the retroviral vector. 6 hours after introducing the virus, scientists artificially fertilized the egg by injecting a sperm from a male. 2 or 3 days later, when the egg had divided twice and become a 4-celled embryo, it is implanted into a surrogate mother. And the Andy was born. It glows when exposed to UV light, emitting a green light. Applications of transgenic animals. The purposes for which transgenic animals have been used indicates the wide-ranging applications of this biotechnology. In medical research, transgenic animals are used to identify the functions of specific factors in complex homeostatic systems through over or under expression of a modified gene, that is the inserted transgene in the progeny. In toxicology, as a responsive test animals for detection of toxicants. In mammalian developmental genetics also, they have the application. In molecular biology, the analysis of the regulation of gene expression makes use of the evaluation of a specific genetic change at the level of a whole animal. In the pharmaceutical industry, targeted production of pharmaceutical proteins, drug production, and product efficacy testing. In biotechnology, as producers of specific proteins, genetically engineered hormones to increase milk yield, meat, egg, and wool production, genetic engineering of livestock, and in aquaculture affecting modification of animal physiology or anatomy, cloning procedures to produce specific bloodlines also, spider silk in milk of goat, that is for biosteel production, then developing animals specially created for use in xenografting, that is organ transplantation. The legal and socio-economic impact of transgenic animals. The production of transgenic animals that potentially can have a deep impact on the livestock and pharmaceutical chains has proceeded much slower than genetic modification of crops. Improvements in animal biotechnology are expected to result in economic benefits for farmers, processors, and consumers. The distribution of this depends on the type of technology, that is a cost reducing technology or a quantity enhancing or quality enhancing applications. Then market structure and competitiveness. In this concentration ratio or suppliers, market power, etc. Then information transparency, that includes labeling and traceability programs, etc. Then price elasticity, consumer acceptance. These are all the type of technologies. Beside the direct economic effects, other externalities, both positive and negative, should be considered in the overall economic evaluation. The interest in genetically modified development in genetic modification development in aquaculture is stronger when compared for the terrestrial animals due to several factors such as better growth rates in fish and improved feed conversion rates that may result in a significant production cost reduction, thus again reduction in the market price. The potential economic impact of the introduction of genetically modified fish could be enormous. The case of growth enhanced genetically modified salmon shows that benefits for producers arising from increased growth rates and food conversion rates. This may lead to a relevant reduction in production costs also and to an increase in gross margin. So at the same time, environmental and human health risks should be deeply considered in the overall evolution of the transgenic fish introduction. Then about biofarming. Biofarming is new territory for the agricultural and pharmaceutical industries and presents novel challenges for government regulators and others. Due to the high cost, the production of transgenic animals such as pig, goat, sheep, and cattle must bring an elevated profit in order to be a feasible economic investment. For this, the production of high value pharmaceutical substances, which correspond to $1 billion markets, is actually the principal and most promising application for animal transgenesis. International bodies such as FAO, that is Food and Agricultural Organization, World Health Organization, World Organization for Animal Health will have an important role in providing forums for neutral discussion and encouraging harmonization on the food sector. Animals such as mice and pigs need to be genetically modified and bred to produce animal for biomedical research, which gives rise to many animal welfare problems and issues. There may be health risk associated with transgenic animals, that is, oocyte extraction from donor animals is usually surgical sometimes. So the animal will be stressed of recovery. Then pregnancy in surrogate mothers is prolonged and heavy painful birth, abnormal fetal development and late pregnancy mortality rates, health problem during life due to respiratory problem and some immune system deficiency, etc. So the continuous breeding needed simply to maintain transgenic lines, well, wasteful of animal life. Transgenic animal produced may not be suitable for research and for further breeding. So simply it would be tortured for the process and is not useful for the research also. These are all some issues arise of transgenic animals constructs. So there may be long-term effects on the environment when transgenic animals are released to the field. Some bioethicists argue that it is wrong to create animals that would suffer as a result of genetic alteration. So some of the solutions we can do is to find a treatment for condition produced or establishment of endpoint for removing animal from a study before problem appears, or we can use embryo or sperm cryopreservation to maintain transgenic line that may not being actively studied. Then we have to develop a guideline to ensure transgenic animal which are used ethically. So finally the conclusion is transgenic technology is a field that is under constant evolution. So many transgenic animals have been successfully created for a variety of purposes, and the prospects are also enormous. It holds great potential in many fields, including agriculture, medicine, and industry. With proper research and careful use, the transgenic animals can go a long way in solving several problems for which science doesn't have a solution till now. Thank you.